The structure of ATP is a nucleoside triphosphate consisting of a nitrogenous base, a ribose sugar, and 3 serially bonded groups. ATP is referred to as the energy currency of the cell, as it provides readily releasable energy in the bond between the second and third phosphate groups. It enables cells to store energy for use only as and when needed. ATP serves to close the gap between energy releasing reactions such as food breakdown and energy requiring actions such as synthesis. Mitochondria is a membrane-bound cell organelle that generates most of the chemical energy needed to power the cell's biochemical reactions. Chemical energy produced by the mitochondria is stored in small molecules called adenosine triphosphate. Enzymes perform the critical task of lowering a reaction's activation energy, the amount that must be put in for the reaction to begin. They function by binding to reactant molecules and holding them in such a way that the chemical bond, breaking and bond forming processes take place more readily. They break down large nutrient molecules, such as proteins, fats, and carbohydrates, into smaller molecules.
What is phosphorylation? Phosphorylation is an attachment of a phosphate group to a molecule or an ion. So, in this process and its inverse, dephosphorylation, are common in biology. Protein phosphorylation often activates many enzymes. So basically, they just it's just the type of attachment, you know what I'm saying?
Okay, here we have 2 substrates, and this is the active site, and here's the enzyme. Energy is first absorbed, and then a bond is formed. Then it goes to an enzyme substrate complex. The substrates bind at an active site enzyme, which then changes the shape to hold the substrates in proper position. And then here we have H2O, which is being formed out, and then here's the connection as these 2 substrates are going in. And then the internal rearrangements leading to catalysis occur. And then here, the product peptide bonds are leaving out, and product is released, which then returns to the original shape, and it is then available to catalyze reaction.
Okay. Phosphorylation transfers energy to enzymes and other proteins. The energy is captured as electrons are transferred from electron donors to electron acceptors, such as oxygen, in a series of reduction-oxidation reactions. The energy released from these reactions is used to drive ATP from ADP.
What?